All right, everyone. Thank you for being here. It is my deep pleasure today to welcome Dr. Nate Lemoine to Cary for our seminar. Nate and I met at the ESA conference because I left my water bottle in a room and they have Cary Institute on them and my name. And Nate is incredibly industrious and looked me up on the website and was like, hey, I think this is your water bottle, and I think we should talk about science. And so now we are collaborators. And one of the big things that you'll see both in Nate's talk and getting to know him is that his background is incredibly diverse. He has lots of different places he's been. So he's originally from New Orleans and laments the fact that he can't find a good po' boy anywhere except for home, which understandable. He did his Bachelor of Science at the University of Richmond and then kind of bopped around for a while, eventually landed in Miami, right? Yeah, FIU, for his PhD, working, right? Yeah, in coral reef ecology. So something we don't get to talk a ton about at Cary often, but is definitely going to come up. And then moved around, did some time at the CERC in Maryland, as well as doing some time at Colorado State University, doing a USDA postdoc fellowship, moving into grasshoppers and grasslands. As you can see from his wonderful title, he's gone off into larger megafauna, and I'm really excited to have Nate here to just talk about this diverse way of thinking about things and kind of finding these through lines in ecosystem ecology. So let's welcome Nate. Yeah. Alrighty.
Okay, thanks. Thanks for bringing me out. I'm really excited to be here and give you guys. This is the first time I've ever given this talk. It's brand new. So you're my test audience to see how this is going to go. And I'm really nervous about it because what Jane didn't mention is that she came out to Milwaukee, was it February? It's like February, and gave like one of the best seminars that we've had in a while. And so now I feel really, really pressured to do well. Not to mention that Evan knows the other faculty in my building. So I don't need Evan emailing them and being like, dude, that dude sucked. And I think if he did that, the other faculty would just be like, so? So yeah, I'm going to talk to you today. There's also fish in this, but I left that off just for succinctness, about grasshopper poop and elephant bones. It's all about consumer-mediated nutrient dispersal. So when you think about nutrient flows through ecosystems, you almost always see it like soil to plants, maybe plants to like the north end of a northbound cow, and then like up into the atmosphere and then rains back down. You never see the line that comes out the south end of the northbound cow back into the ecosystem. And that's really important because they recycle elements just as well as bacteria. The thing is, like, people like Jane came up with fancy words like nitrogen mineralization and phosphorus cellulization to cover up the fact they're really studying bacteria excrement, but I study animal excrement because we are like reducers and recyclers of material, especially of material that's bound up in plants, which is typically refractory. That's our job, is to take this material, recycle it, mineralize it, and put it back into ecosystems. And often these ecosystems are nutrient limited. And so it turns out that animals, this consumer-mediated nutrient dispersal is incredibly important and a huge component of ecosystem nutrient flows. So the top, from your angle, top left graph, if you actually just look at the second bar, which is nitrogen input into coral reef ecosystems from carnivores, snappers, grunts, those kind of things, is like 10 to 20 times higher than any nitrogen input from an abiotic source. So it's a huge input of nutrients into the system. If you do some back of the envelope calculations for grasslands, you can see that mesic grasslands like Hays, Kansas, Konza Prairie, I didn't put it up there, but Spring Green, Wisconsin, the nitrogen input into those soils is way bigger than anything that comes from mammal inputs, abiotic sources, you name it. And then elephants, we're not going to talk about elephant poop or pee, although that is likely a huge input into systems. We're going to talk about the elephant itself. So you may or may not know African bush elephants, the biggest terrestrial mammal left existing, weighs about, on average, 4,000 kilograms. Of those 4,000 kilograms, there's 800 kilograms of carbon, 300 kilograms nitrogen, a lot of phosphorus. And so a single elephant put into a single spot more nitrogen than accumulates in a century of atmospheric deposition. And if you actually extrapolate across the park for Kruger National Park elephants, mortality, you get 480 metric tons of carbon being put back into the ecosystem every year. You get 180 metric tons of nitrogen coming out of elephants, just their bodies being put back into the park every year. And so I'm going to make the case that consumer nutrient dispersal, what I'm going to call C&D, is hugely important in all these systems for structuring primary producer communities, for stabilizing soil function, and for enhancing savanna diversity. And we're going to bop around through a bunch of different ecosystems. I'm glad I came and saw Evan's talk yesterday because it's a really good setup for hard breaks and then turning lefts into different ecosystems. He stayed in tropical forests, so it was just like a nice soft launch for what's about to happen now. So we're first going to talk about the structure of primary producer communities. This is work I did largely back in my PhD, but I'm actually getting back into through the LTER network now. This is what used to be a coral reef. So this is from Davies Reef in the Florida Keys. It used to be a coral reef. This is sadly what most of the Keys look like these days, is mostly just rocks. There's no coral left. But the first thing you should notice about this reef is just the sheer amount of fish biomass. It doesn't matter if there's coral there or not. There's hundreds, if not thousands, of fish. And for those of you who don't know your fish, these are almost all carnivores. So the yellow tails are schoolmaster snapper. The other ones are grunts. They're all invertebrate eaters or small fish eaters. So they're all carnivores. They're all recycling nutrients. There are a few parrotfish in here. And so you got hundreds, thousands of fish swimming around. They're constantly excreting. They actually excrete ammonia. As soon as it hits the water, it turns into ammonium. And so you have this massive constant dump of ammonia. You can actually see here, there's your beach sand coming out the bottom end of a parrotfish. So there's actually on camera. Tons of nutrients being dumped onto this reef by these, and these schools are pretty resident. This reef almost always looks like this. So the question is, what does this nutrient input by all of these fish do for a coral reef ecosystem? There's reason to believe it might not do much. I mean, you're in the ocean. Some of them are a meter or two off the seafloor, so it might just wash away. Is there any reason to believe that some of this actually makes it back into the primary producer community? So I'm going to make the case, hopefully, that consumer biomass drives nutrient availability in these oligotrophic systems. Then this nutrient availability actually is taken up by the primary producers, the algae in the system. And then that increased nitrogen availability in the algae can actually drive herbivory and bring in herbivores and attract herbivores. So let's start with how we do this. There's two different scales at which this happens. So this is work done by my friend Andy Shantz. Evan and I talked at dinner last night. I actually did a lot of the statistics for this, but didn't wind up on the paper because it was more like him turning around in his office chair and be like, how do I program this in R? So I know these data really well, even if I'm not on the paper. And he didn't even put me in the acknowledgments, which, whatever. And he expects me to be his best man. We'll see about that. Okay, so this is within a reef. This is a reef called Pickles Reef, called that because there's a ship that sunk. You can still find the pickle barrels it'll come back. Pickle barrels it was carrying. And what you see are these dendrogyra colonies, pillar corals. And what you see is some of these don't have a ton of fish, not a lot of fish schooling here, and some of them are home to these large fish schools. And this is really consistent. The fish will actually repeatedly return to the same structure. So you have some sites within a reef that have low fish biomass and some that have high fish biomass. So we can study this process within a single site. And then what I actually did participate in physically was going all over the Florida Keys from top to bottom and surveying reefs that vary from very low fish biomass to very high fish biomass, and looking at nutrient excretion in this scale. And so if we actually look at the within reef scale, this is a video of some grunts schooling, just to remind you of just how big these fish schools can get. If you actually measure nutrient excretion into the water column, what you get is what you expect. You get lots of nitrogen coming into the water column in areas with lots of fish, very little nitrogen coming into the water column when you don't have a lot of fish. And the same is true for phosphorus. The availability of phosphorus goes up by 3 to 5 times when you have all these fish recycling nutrients in this area. So the absolute amount of nutrients is driven by the consumer biomass in the system. What we've been working on recently also through the LTER network is aggregating data from all marine ecosystems in the Pacific. So we have kelp forests, northern oceans, Maria, all these marine data. This doesn't look like that many data points, but it's actually the end product of like 1.6 million observations that Mackenzie White did an amazing effort synthesizing. And so there's a lot more that goes into this, but what we also found is that the stability of this consumer nutrient excretion is also determined by the stability of the biomass of consumers in that system. And it also has to do with synchrony. So the more synchronous the populations are, the more unstable this nutrient excretion is. So what you really need is asynchronous population dynamics to get stable nutrient dynamics. And this was done by the LTR network. I tried my hardest to find a picture of all of us together, and it shows you how bad I am at pictures, is that the only 2 pictures from my entire time in California are me and Darren's dog hanging out. That's all I got. But there were a lot of people that participated in this. People like Mac, Joey, Jake Allgaier, and a bunch of other people really drove this, and I just came in at the end and did some of the modeling. So the 2 points here is that stability of nutrient excretion driven by stability of consumer biomass, and the amount of nutrient excretion driven by the amount of consumer biomass. So consumers are really driving nutrient availability in these systems. Does this actually translate into higher nutrient availability in algae? It turns out it does. So these are the data from the within reef comparison. If you sample what's called Diota, which is just a little brown algae, from the high biomass sites, you see that it's got significantly more nitrogen in its tissues than it did at the low biomass sites within a reef. And then if you do the same thing across all of the reefs, you find that as you have more consumers, you have more nitrogen in the water, you have more of that nitrogen being taken up by the algae in those systems. And fish, somehow, I still don't know how, can tell. So when you have higher nutrient-enriched algae in these systems, the fish will come in and preferentially feed on it. Parrotfish and acanthurid bite rates were about 3 times higher under the pillar coral colonies with lots of fish than they were outside of it. And this is a video to kind of show you what I'm talking about. You can see this highly structured environment with lots of fish biomass. The parrotfish, which are these blue ones, are just really hammering the reef. They're really interested in like tiny little algae tufts that grow on this reef because they are so nutritious. And so there's a bonus one here because this enriched algae concentration can bring in more herbivores. And when you get more herbivores, they actually structure the community. And so there's a back and forth here where if you do the community composition from the high fish to low fish biomass areas, you see that there's lots of what CCA is called crustose coralline algae. It's like the pink stuff that grows on top of reefs. It has no vertical structure to it whatsoever. And then turf algae is what you think of when you think of like an algae-enriched reef. It's like the little tufts that stick up. When you have a lot of carnivore biomass, you have a lot of crustose coralline algae, and that's kind of opposite what you expect. Like if you see a reef awash in nitrogen and you think that there's just fertilizer being dumped on it, you should get lots of primary production and lots of algae growing. And you just don't because you have parrotfish like this that come in and preferentially feed on all those nutrient-enriched algae, and they keep, like if you look at this reef with all this nitrogen, it should be covered in algae given how much nitrogen there is. And there's almost none because you have parrotfish like this one and this one up here. You can watch them just repeatedly hammer the reef over and over and over again. So not only does the high nutrient availability attract the fish, but then the fish through high herbivory rates will actually control the structure of the primary producer community. So there's a feedback that goes both ways. So ultimately we have consumer biomass leads to more nutrient availability. Those nutrients get taken up by the algae, and then that high nutrient availability in the algae attracts herbivores, and by attracting herbivores, they then control the community composition of the algae themselves. So it's like a really tight story that gets told in this reef system. So the idea that consumer-derived nutrients can actually structure primary communities appears to hold, at least in coral reef systems. So now we're going to do a hard left turn and say, okay, so that's all about population and communities. What about ecosystem function? And this is something that I've been working on with Jane quite a bit. So we're going to skip over lots of ecosystems from coral reefs to the Midwest prairies. So this is our study system at Spring Green. It is one of the largest, if not the largest, remnant prairie in Wisconsin. It's dominated by big blue stem, little blue stem. You see a burn break, so they do burn regularly. So this is unburn, this is burn. And one of the big differences you see between this video and the video I played of the reefs is you see a complete lack of animals, right? Like the fish video had hundreds of thousands of fish swimming around. You don't see any of that here, but it does not mean the animals aren't out there. You can't see them because, my camera's only so good. So there's actually hundreds or thousands of grasshoppers all through here, and they're all doing their thing. They're all taking up refractory plant material, processing it, depositing it in frass as more labile material, and dumping that into the soil. And so then the question is, do these herbivores actually do something through converting slow process detritus into fast process detritus? Does that actually affect the soil properties in this system? So, and this is just to remind you just how much nutrients the grasshoppers are putting out. So it's not just like a little bit that they're dumping out there. It might be a little bit in any one space and time, but they're constantly moving back and forth. And so over the course of the whole prairie, they're actually putting quite a bit of nitrogen converted back into the soils. So we do this. Damn it. Right, so the story I'm going to tell you is that these herbivores destabilize microbial communities. And when they do that, they decouple respiration from water availability. And when you decouple respiration from water availability, you actually wind up stabilizing carbon use efficiency and ecosystem function to fluctuations in water availability. So ultimately, what I'm going to argue is that grasshoppers are insanely important for stability of ecosystem processes in grasslands. So this is something Jane has been helping me on through data analysis and conceptual thinking. So what we've done is we've set, now I can say this, that we do this by actually setting up drought shelters in these environments to manipulate soil moisture availability. And then within each drought shelter, we have cages that keep herbivores out, cages that let herbivores in. We go out there, you can actually see, this is Claire, that's her way back there too. So we actually go out 3 times, once a month over the summer, collect soil samples from all of these plots and measure respiration. We measure 16S abundance. Jane and I have started doing metagenomics, but we haven't looked at any of that yet. We measure nutrient availability, the whole suite of soil function that we can measure. And this is 2 of my undergrads who I couldn't have done this without. This is Claire, that's Maggie. What we do is we take these samples back to the cafe that's nearby, and they really like to let us sit on the porch and just like set up our lab and measure all the respiration right there. And we buy sandwiches and stuff from them, so it's like a nice give and take. So I also really have to thank, before I go any further, my undergrads, Anesti, Maggie, my lab manager, Michelle, and then 2 more undergrads, Diego and Claire. They're responsible for a lot of the data that got collected. They've been out in some pretty crappy conditions. This is us out. This looks like it's not that bad, but it's like 40 degrees and pouring rain. We actually had to call it for safety reasons and like go to Walmart and buy new clothes. We were so wet and cold. So like I really owe a lot of what's been done to the fantastic undergrads at Marquette. So the first fact, or first thing that I'm going to propose is that herbivores destabilize microbial communities. So, and by that I mean they make them more variable throughout the year. So what we did was we actually did 16S sampling on samples from early, mid, late summer. And we found that when you remove herbivores from the system, so you go from this orange box to this blue box, the communities become more stable. So these are less stable than these communities. And which is kind of a weird result because we always think of stability as being this great thing for ecosystems. And so you think, well, if the herbivores are destabilizing the community, that can't possibly be good. And one of the things to notice is that this isn't a huge difference on the relative abundance scale, but if you look at it, if you assign these into functional groups based on copiotrophy and oligotrophy, you see a huge difference in the stability of those functional group breakdowns. So when you remove herbivores from the system, you get a lot bigger shifts over the summer from oligotroph to copiotroph and then back to oligotroph again. And when you have herbivores in the system, when you remove herbivores from the system, you're kind of stuck with one functional group. And when you have herbivores in the system, you're going back and forth between functional groups a lot more readily. And so that actually turns out to be a good thing because it allows the microbial community to adapt to the changing conditions over the course of the summer. So if you actually look at the relationship between respiration and soil moisture, When you remove herbivores from the system, you get a much stronger relationship. So what this means is, if you think about this in terms of sensitivity, the sensitivity of microbial respiration without herbivores is much higher to soil moisture than when you have insect herbivores present. This is relatively, it still matters what the water content is, but it just doesn't matter as much when you have herbivores present. And that's because herbivores are allowing this community to shift back and forth to account for these changing conditions over the course of the summer. And ultimately what that does is that stabilizes respiration. So over the course of the summer, as rainfall is going up and going down, when you take herbivores out, respiration is going up and going down much more extremely than when herbivores are present. It's still fluctuating a little bit, but just not nearly as much when you have insects present. So it stabilizes this over the course of a growing season. And then because respiration in our system, we actually did qPCR and quantified it and calculated microbial carbon use efficiency. And so it turns out that when you stabilize respiration, you are ultimately stabilizing microbial carbon use efficiency as well. So it turns out that grasshoppers are hugely important for stabilizing below ground function, but they do it in a surprising way. They do it by destabilizing microbial communities and allowing those communities to adapt to the conditions as they change more readily. And that can then in turn stabilize the ecosystem function, which is also really important for how we think about stability and whether it's a positive or negative thing, because sometimes you might want a community that is less stable and more able to adapt to changing conditions in order to maintain a set level of function, which is part of another working group that I'm in right now. So I've told you now that on coral reefs, consumer nutrient distribution can structure primary communities. I've simplified the story a lot, which we cannot talk about later, but I've suggested that consumer nutrient distribution can stabilize ecosystem function in grasslands. And now I want to talk about how consumer nutrient distribution can actually enhance savanna diversity. So we got to talk about elephants. It's one of the things I'm probably known best for. Everybody knows that elephants maintain savanna communities. Like, for those of you who are here before and listening to the movie play, like, we know that savannas don't exist without elephants, and we know that it's because they do this kind of stuff, right? They go through and tear down trees. I like this video because the elephant kind of backs off for a second and is like, just push. It comes down. And then all the other elephants get really excited, and they're like, all right, we can go in now. So they do this kind of stuff constantly. So without elephants, you would have a much shrubbier savanna. So we know that living elephants are amazingly important to maintaining savanna composition and distribution throughout Africa. We also know that elephant carcasses are hugely important food sources for a lot of animals in the savanna. So this is an elephant carcass we found like 10 minutes after it died. Went out the next morning and actually got to sit there and video all these cute lion cubs going in and feeding. So you can't see them, but this pride of lions about 15 strong. So there's actually some over here, there's some back here, there's a whole bunch back here you can't see. And the other thing you can't see is that behind us is like every vulture from Botswana and South Africa is sitting in every tree behind us. I've never seen so many. So yeah, we know that elephant carcasses are really important food sources for lots of animals out there. But also, keep in mind how many nutrients are about to be just dumped into the soil here. So there's reason to believe that there's a huge ecosystem impact. And the whole point of this process is that elephant bones, we thought, were too big to be moved around easily. And so there's the thought that they stick in one place, and so there's this long-term nutrient enrichment that you get from elephant carcasses that you don't get from smaller animals that are just like scattered, the bones are destroyed within 6 months to a year. Elephant carcasses you can actually find 8, 10, 15 years later, ostensibly. Yeah, there he goes. So the thought here is that elephant carcasses increase nitrogen and phosphorus availability in the soil. And the key is we know they do immediately, but we think that it happens for 5, 10 years afterwards. And we think that that means there's these long-term patches of altered productivity and altered grass community composition where, like, if you're walking through the savanna and you see like a little patch of grass that looks different, could be a potential legacy effect that an elephant died there 5 years ago. And then those patches, by being enriched in nutrients, then attract all kinds of grazers like impala, zebra, and all kinds of other things come in. And it's a hotspot that then becomes a positive feedback because you have all these grazers that come in, they eat the grass, and they themselves pee and poop all over the place, and that just kind of maintains itself. So that's the hypothesis, is that this happens for over long time scales. So how do we do this? We fly into Kruger National Park. It's difficult to get a sense of the scale here, but it's about the size of New Jersey is the common reference. So Skakusa is down here, and so that's where we fly into and we're based out of, and then we travel all up and through the park. The park is nice enough to share the georeference locations of every elephant carcass they have with us, so we know exactly we know roughly where the elephant carcasses are. Sometimes the elephant carcasses, geopoints are where the ranger got back to his truck and not where the carcass is. There's some hit and miss there. And not all elephants are nice enough to die along the paved roads for us, so we also wind up down things that are ostensibly called roads and have to do a lot of off-road driving. You can see my lab manager here just really enjoys doing this. And this is my grad student, Nova, in the front seat, who's also seems to be having the time of her life. And then once we get to these sites, often we can only get within 1 kilometer or 2 of the elephant. So then like the 15 of us have to get out of our trucks and we have to walk through the savanna. So this is us walking through the savanna with, wildebeest, zebra, it's hard to see in there. And I have to trek 1 kilometer or 2 through the savanna to get to the sites. And then when we do, we've carried all this equipment out there. So we identify where we think the center of the carcass was, take every measurement we can think of. We do grazing, we do plant decomposition. This is a DPM, which is a disc pasture meter. It's a measure of plant biomass. You just drop it, and obviously the lower it goes, the less plant there is. We take soil cores and do soil respiration. We're doing microbial community analyses, enzyme analyses. So everything from soil ecosystem function up through plant communities, up through like large herbivore behavior. We have game cameras up and stuff to watch like animal behavior studies. So this last time in March, we were pretty good. We found about 32 carcasses that we were able to sample, which is pretty good. So I can't go on also with this without giving a shout out to the hardest working team as part of this project is my team, the soil team. We have to carry the heaviest equipment. We have to get up in there and get the dirtiest samples, the stinkiest samples. We have to carry all this stuff several kilometers often, and then once we get back into the lab after long day, we have to spend another 6 to 8 hours in the lab running respiration. So 3 weeks of 14 to 16-hour days constantly mixed between field work and lab work is a lot to ask of people, but they did it. This guy's not even on my team. This is Andy, the guy who expects me to be his best man. He just came down for shits and giggles, and we're like, we're gonna put you to work. And he's like, all right. And he got to it. So really, like, this is Andy, this is my grad student, Nova. This is a local college student, Ali Maori. This is me over here. This is our team. My lab manager, Michelle, pops up here. Couldn't have done this without them. All right, so let's talk about the increased nutrient availability. So let's look at some of these graphs. On the right, you have granite soils, which are nutrient poor, and on the left, you have basaltic soils, which are nutrient rich. And then you have the lines colored by elephant age, so 2 months, a year, 3 years, 5 years. So what we see on granite soils, first off, the young elephants have 500 milligrams per liter of nitrate, which is like an insane amount. And that's the average. Some of the numbers are up in the, if we were to plot the points, it'd be like in the thousands. But the interesting thing is like, yes, it's elevated 2 months after the elephant dies, but it's also elevated still a year after the elephant dies. And it's also elevated 3 years after the elephant dies. And that's consistent in both basalt and granite soils. Even some evidence that 5 years after the elephant has died, nitrate concentrations have remained elevated. The pattern is even stronger for if we look at plant available phosphorus. Over here you see elevated phosphorus from 60 days, a year, 3 years, like insanely elevated levels of phosphorus, because the bones stick around mostly. We'll go over that in a second. And they leach over time phosphorus into the soil. So the longer that the bones stick around, the more they're leaching into the soil. And what we see in the basalts is actually the opposite pattern, where the older the elephant carcass is, the more phosphorus has accumulated under the elephant. And I put this here, and we think that this is because there's 2 different communities of wildlife on these 2 different soil types. On the granites, you don't have a lot of lions, and on the basalts, you have a lot of lions. And if you've seen The Lion King, you know they don't get along with hyenas. That's actually true. So when you have a lot of hyenas, they will come through and disperse the bones in 2 weeks. You won't even know the elephant carcass was there. The bones are like scattered 50 meters. You may not even find most of them. On the basalts, where you have a lot of lions, there's much less hyena activity. And so the bone fields will actually stay much more intact for long periods of time. So we think that the wildlife communities that are on the scavenger communities on these two different soil types are driving a lot of the phosphorus dynamics by the way they interact with bones. I also want to point out that it's not just the scavengers that interact with bones. We know other animals do too. So this picture shows, this is an elephant carcass that's about a month and a half old. This is where we found it 2 weeks before we took the picture. So some animal came in, grabbed the entire carcass, rotated it 180 degrees, and then dragged it 20 meters off from where we originally sampled it. So, which also raises questions from our sampling design too, of like, where do we sample when we go back? Do we keep sampling here? Do we move here? And if you wonder what animal is big enough and strong enough to do that, it's these assholes who know we're out there, and they know that we're doing this, and they throw our stuff into the woods, and they smash our stuff up, they steal our game cameras, and you can see this elephant caught on a trap, like just tossing bones around for what is apparently no real reason. So we know that elephants will go back out to these sites. We know they kick the skulls around, like we can go from site to site and go back, and by sometimes 50 meters, we've seen them kick the skulls around. We don't know why they do it. We're actually doing some behavioral studies and found they're way more aggressive towards foreign objects when they're on carcass sites, so they do seem to care, at least to some extent. So whether these nutrients alter primary productivity, we actually expected to see hotspots of primary productivity as we go up, and that's actually not what we see. What we see is that young carcasses have a lot less primary production to the point where there's almost none. That's fine. You just dumped 300 kilograms of ammonium onto the soil and poisoned it, killed everything. But what's amazing is that the primary production on these carcasses pretty much stays suppressed, and in fact becomes even more suppressed as the carcass gets older. And that's true on granite soils. On basaltic soils, it's just kind of always suppressed, but even up to 5 years after the elephant has died, you just have less plant biomass on top of these carcass sites. And the same is true with species richness. So you have fewer plant species and less biomass on top of the carcass sites for up to 5 years after they've died. And the other thing, and I have to credit Dave and Helga here, the two South African collaborators who did all the plant work. Like, this is a hard task to go out and do species comp across the park. The other thing we see is that if you look at this, you see that the carcass sites, the 0 meters out from the carcass, has a very distinct plant community from the matrix surrounding it. So we know that the plant communities are different. One of the things to pay attention to here is these orange dots, which are kind of like 15 meters away, represent the control conditions. You see a lot of separation, and the shapes are soil type. So you see like basaltic soil plant communities over here, granitic plant communities over here. By the time you get to the carcass site, they've all converged on the same thing. The carcasses are almost always simply Uricloa and Panicum, and that's all that's there. So you have a lot of convergence in community composition on top of a carcass site. And we think it's due to a physiological tolerance to ammonia toxicity. And so my undergrads are actually, they call themselves the Odd Squad. They're actually doing this kind of ammonium experiment in my lab right now to go along with this. We're poisoning 9 different species of African grasses and seeing which ones are the most tolerant to it, to match this. So what happens, and the reason that this biomass stays suppressed for so long, is because grazing keeps going up. So grazing stays elevated on carcasses continuously for up to 3 to 5 years afterwards. And so because these are hotspots of grazing, it's not surprising that primary production is so much lower. It's because this has become a place where grazers will congregate to feed on the grasses. And I haven't shown it, but it's because the grasses are more enriched in nitrogen, phosphorus, and all kinds of calcium and other micronutrients. And we've done stable isotope work, and we've known that those nutrients are coming from elephant carcasses based on the stable isotope work. So the elephants are feeding into the plants are getting more enriched, which is causing more grazers to come in, which is now changing the plant community and reducing biomass. The grazers, these are fun videos because the two you're about to see are like of animal behavior that is not well, if at all, documented. So we've also discovered through game cameras that the grazers will come in for what's called osteophagy. So I've never seen this before. This is a very startled African buffalo that was caught about licking a bone. So it turns out that the herbivores themselves will come back to chew on the bones. And sure, you can look at this and be like, well, you didn't actually see him eating the bone. He could have just been sniffing it. Fine, whatever. You can be cynical, and I'll raise you 2 giraffes who we did actually see eating the bone. So this is a giraffe actively chewing on what is the jawbone of another giraffe. This is a giraffe that came in and decided it was just going to go to town on what I think is an elephant femur just hanging out. And so herbivores will not be attracted just to the primary productivity in these sites, but they will come back to actively feed on the bones. And we think that that's like it's like putting a salt lick out there, right? So we think the herbivores really like this. Again, this behavior seems to be more common on the basalts. And if you remember, that's because there's probably less hyenas on the basalts, and so there's more bones left up there, and there's less competition for them with the hyenas. The hyenas will take the bones off. We have game cameras of the hyenas carrying them around like a dog does. Like they just like, I'm gonna pick up my bone and then go over here away from all of you and sit down and chew on it for a while. So I think this is related to the availability of bones for them. And the other thing we've done, and Evan reminded me of this because he did drone flights the other day, and I was like, oh yeah, we did that too. So we actually went out and did drone flights over one of our sites with Andrew Davies from Harvard. And so you can see this is the kind of imagery we get back. We get back 3-dimensional structure. And so you actually see right here, this is the elephant carcass, and you can see the depression it makes in the vegetation height around the carcass. And that's kind of cool, we know that. What I think is really cool in terms of grazing is if you actually look at it, you can see our carcass sites here, and you can actually, if you squint, see game trails. Like Andrew's drones are good enough that he's picked up, they're 1 centimeter by 1 centimeter resolution, and he's picked up game trails. And you can see all the game trails converging onto this carcass as herbivores continually come back in to feed on it. And I thought this was a pretty cool result. And so we're hoping to get Andrew back out and to do this on more carcass sites. This was kind of a trial run. All right, so for elephants, we have elephants increasing soil nutrient availability. We have that soil nutrient availability altering plant biomass and plant community structure, and it does so through maintaining grazing, right? So the elephant nutrients enrich the plants, the grazers get really excited about it, they come in and they keep repeatedly coming back over years and years and years, and so you get a totally different plant community composition that we can now identify, like just walking around, the legacy effects of elephants. And if that sounds familiar, it's because you've heard this story before. It's the same exact story I told you about coral reefs. And so you have the same kind of loop happening with consumers dumping nutrients into an ecosystem, feeding the plants and enriching the plants, and then attracting herbivores and altering the community composition. And you have it happening in coral reefs and savannas in pretty much the same way. And so I thought that this was kind of a neat way to tie all of these stories together. And so ultimately, I think that consumer nutrient distribution structures plant communities, it stabilizes soil function, and it enhances savanna diversity, and it's doing it through similar mechanisms in all of these different sites. Okay, now I'm gonna give you guys a bonus. There's a 4th one that I didn't talk about, and this is more work that I've been doing with Jane. Consumer nutrient distribution is not always good. I've been pitching it as like this good, important ecosystem service, but there's 7 billion of us dumping nutrients into wastewater treatment plants constantly, and what does that do to ecosystems? And just as an aside, like I was trying to be gender equal and find a stick figure representation of a woman, and the only one I could find was of her having a really bad day. So I tried. But it turns out men are just way easier to depict doing those things. So the reason we're interested in this is an idea that my grad student, Novali, well, Jane and I's grad student technically, had when came to me and said, I want to look at artificial sweeteners. And the reason that this is important is because as obesity rates go up, more and more of our products are containing some form of artificial sweetener, whether it's sucralose, ACE-K, aspartame, some kind of artificial sweetener is getting into all of our diet products. And the reason that that is important is because they are by design not metabolized. So if you drink them, the whole point of them is to not be metabolized and to come out the other end. So what Nova was interested in is what role these artificial sweeteners may have on soil health, because they do get into our soils. She had that research. So you put artificial sweeteners in our food, they're really common in livestock feed. It doesn't seem to do anything. The animals don't seem to care, but people still think it does, so they still dump it in there. And by default, they're not metabolized. We either throw it out, put it through the toilets, winds up in our wastewater treatment plants, which you can't treat out something that bacteria won't metabolize, so they just continue on and wind up in our aquatic systems by way of kind of wastewater treatment. And if you're fertilizing your agricultural field or you're dumping kind of gray water on your lawns, you are dumping artificial sweeteners on your lawns and on your agricultural fields. So NOVA actually found reports from Dane County, which is where Madison, Wisconsin is, that found detectable levels of all of the artificial sweeteners in the former residential area that had been abandoned for 10 years. They were still in the soils in really low concentrations, but they were still there. So her question was, how does this impact soil function? So she designed this great experiment where she just collected soils, sieved them, did a really simple bottle experiment where she did low, medium, and high dosages of multiple different sweeteners and just measured soil respiration, and then also did ecoplate analysis to look at substrate usage. And so just with the question of, do realistic levels of sweeteners in soils impact soil health? And it turns out they do. So the first thing to notice is that the levels of sweeteners is like 1 microgram per liter, 5 micrograms per liter, insanely small concentrations of artificial sweeteners. And it doesn't matter if it's ACE-K or sucralose, you're getting a 30 to 40% reduction in soil respiration when you add these artificial sweeteners into your soil. ACE-K gives us this nice like low to high line. Sucralose is doing its own thing. I don't know how to interpret that, but what I do know how to interpret is that it doesn't matter what it's doing. What matters is that as you add it into the soils, it's suppressing respiration. So you have to think about what this means as we're spraying artificial sweeteners, laced soil, water all over the soils throughout the Midwest, that we are unknowingly suppressing microbial activity by about 30 to 40%. And she also did some substrate use stuff and found that you're actively changing the community composition. We do have metagenomics, and Jane's been waiting on NOVA to Nova's in her 2nd month of grad school, so she's a little overwhelmed. So we've put that on hold until she settles in. But we do have metagenomics on all of this to look at relative abundance and gene expression in these samples as well. So we think this is going to be a pretty solid paper. But one thing we do know is that when you add both sucralose and ACE-K to soils, you get very different substrate usage by way of ecoplate measurements in these soils as well. You move from like a carb and acetic acid dominated group to much more polymer and phenol dominated groups when you add artificial sweeteners to these soils. So, basic message, consumer nutrient distribution, hugely important, whether it's us peeing into toilets and then going out and fertilizing our land with that, fish, elephants, grasshoppers, you name it. I think this is a critical component of ecosystem function and biodiversity, and I think it's something we need to consider more deeply as we think about heterogeneity on a landscape, right? There's a lot of hidden heterogeneity that is caused by consumer movement through the landscape that's not picked up by other processes. And so with that, I will end this talk, and I told you she was there. And I will take, that's my dog, Sasha, and I will happily take any questions you might have. This goes for a while too, so don't feel like you have to stare at the whole thing.
All right, thank you so much. So we'll take any questions from the audience here first, and I'll just remind you to hopefully rephrase the question for them, and I'll try to remind you again when you inevitably forget.
Right.
Yeah. Lauren.
So in the African ecosystem where you're looking at your elephant carcasses, how big of a role do you think the scavenger community is playing on the results that you're getting? Like some areas probably have a lot more vultures than other areas. Presumably those are consuming bulk of the meat over the course of the entire carcass.
Okay, so the question was, how big a role do scavengers, things like vultures, play on our results? And we think that they're critically important, especially their spatial distributions throughout the park. How much of the carcass is consumed by vultures versus hyenas versus lions? I don't think we know, but we do know that hyenas will come drag the bones around and spread them out. And so we think that when we're seeing weak effects of like the bone field, it's because they've been dispersed by scavengers compared to parts of the park that don't have high scavenger densities. Or maybe the scavengers, like in the North Park, where I showed you the lion video, the scavengers are almost all lions who aren't gonna move the bones around, or vultures, like the hundreds of thousands of vultures who are not big enough to move the bones around, so the bones stay more intact. So we think that that really is like probably one of the strongest determinants on how consistent the elephant carcass signal is through time. Yes?
Really nice talk. Can you talk a little bit more about like the scale at which this heterogeneity is important?
Yep.
You know, in sort of internal recycling of nutrients versus actually, you know, moving nutrients from one place to another. And yeah, so what spatial scales maybe in some of these different systems
So,
is really important?
yeah, so the question is, what spatial scales is consumer nutrient recycling important? In coral reefs, it's clearly the reef scale because, you know, like there's variation within reefs as Andy's stuff showed from coral head to coral head, but by far the biggest variation is from reef to reef in terms of consumer biomass. And so, you know, it's really important, and you know, a reef is anywhere from 100 meters long to, you know, a kilometer long. It depends on the reef, but that's the scale at which changes in consumer biomass happen. The elephant carcasses thing is 15 meters, right? But elephants, you know, the idea is that there's 20,000 elephants in the park, 6% mortality rate. So if you imagine that's about 300 elephants dying every year, all through the park. And so what we think is like, yes, a single elephant carcass is small, but there's flickers of hotspots. There's 300 of them a year scattered all throughout the park. So one of the parts of this project that we hopefully get to is like a spatial modeling to see like, is this actually a landscape level effect that persists through time? So we think that it's important, but we don't know the scale in African savannas just yet. Yeah, Evan.
Really cool work. I was curious, you mentioned some micronutrient stuff, so you might be like already very far down this path, but there are some instances where the stoichiometry of the primary consumers is disconnected from the stoichiometry of those consumers. And I think about this,
Yep.
like tropical regions where you have like salt licks and everything, because something like sodium won't be in plant tissues, but it's required for nerve function.
Yeah.
I could see this being really important for some of that because you have your consumers directly like, inputting in certain areas. Have you gotten into those types of micronutrients that might be typically reduced in primary consumers and then maybe elevated in these locations?
Yes, so we do have a paper that's in review in EGU Sphere, I think. Kind of a strange review process, but so it shows that there are like certain ions like that, those microions that are elevated. So things like calcium, potassium, zinc tend to be elevated in these plants. And speaking of scale, one of the proposals I have in right now is talking about sodic soils, which if do you all have, anybody heard of sodic soils? It's a salt-enriched soil because water will move downhill and then evaporate. And so it's this smallish, probably about the size of this room area on the landscape that is highly enriched in sodium, calcium, those things. And it attracts herds of buffalo, herds of zebra because they like it. So we know that the salt and all the micronutrients play a huge role. We do have all of those measurements. And one of the things we're also really interested in is that the switch in stoichiometry through time, because as you see, N starts out really dominant and then phosphorus takes over. And so we actually see changes in plant stoichiometry going from high N to P to low N to P ratios through time. And so the stoichiometry itself is also dynamic. Yes?
Thank you for your talk. So I was curious about the reduction in soil respiration and like how do you think that will impact like overall carbon fluxes, knowing that soil respiration is like a huge part, like carbon fluxes and carbon emissions.
So the question was all about what, and this is for the artificial sweetener work?
Yes.
So how will the artificial sweeteners impact soil respiration in terms of carbon flux? And I'm gonna cop out of that question because I have another undergrad who's studying the effect of herbicides and showing that if you spray herbicides on soils, you double soil respiration. So the effect is gonna be contingent on like all of this other crap that we're piling onto the soils at the same time, right? Like Nick's work showed, like he put in glyphosate into these bottle experiments and respiration doubled. And so really is gonna depend on the other things that are in the water. I don't know that anyone can reliably answer that question right now. Yes?
Hi, great talk.
Thank you.
I was curious about, in the very beginning, you had a graphic of the elephant. You'll have to remind me, it was like 800 carbon.
Yep.
How do you get to that number from something like the average weight of an elephant?
We used ecological stoichiometry by Sterner and Elser, and their like average makeup of an animal in terms of percentages, and just like did the percentage of a 4,000 kilogram elephant.
And so that's presuming that's what's left there, and then it's consumed by scavengers or consumed by
Yeah.
whoever. And then, so it's also sort of an unresolved question, how much of that ends up in soil?
Yes, so yeah, the question and correct point is that it's also unresolved, like of the 800 kilograms carbon that gets dumped by an elephant, how much of that carbon is dispersed into scavengers and how much actually goes into the soil. Yeah, unresolved, because it's going to vary with the scavenger community and stuff. Yes?
It's just so striking, like the primary production versus the animals that are eating it in the reefs.
Mm-hmm.
And like how that system is able to maintain so much biomass of animals. Like, I don't know what my question is. Can you just talk a little bit about that? And I mean, so basically like the algae are growing, but like immediately it gets eaten.
Uh-huh. Yeah. But but most of the nutrients, so when you looked at that video, there's a reason that 99% of those fish are carnivores. And so like you have some parrotfish in that video, but there's so little algae, the parrotfish are like fighting for what's left. Those carnivores are actually migratory, so they school over the reef in a day. And then at night, they'll disperse into the surrounding seagrass bed and pick off, like, you would not want to be a little crab or crustacean or anything anywhere near that reef within like a kilometer, because at night is just terrifying. And then they all come back to the same reef during the day. So that population is maintained in that small space because they go feed elsewhere and then come back.
So does having that like hot spotted nutrients then also structure the community of the other animals that they're eating?
Open question. Don't know. We're reef people, not seagrass people.
Question we have online is so there's a homogenization of the plant community. Is there also a homogenization of the soil nutrient dynamics in these kind of elephant carcass hotspots?
I think that is unresolved because we have not looked at those data. Is how, like, we have the data, nobody has opened them or graphed them yet, so maybe. Way back.
So I'm wondering if under those furry scavengers, there's a huge change in the insect community around the carcass sites, and then thinking about your grasshopper grassland study, which I understand are not carcass eaters. Is there an insect stored in the soil microbes?
That's the
elephant as well?
That's the entire reason I'm on that project. So the question is whether they're insect responses to the carcass. And I will say I've had a expletive of a time getting those insect data. It's just been real hard. So we do see preliminarily some slightly different grasshopper species that pop up on the carcasses as opposed to off the carcasses, which tracks with termite mounds, which is known. But that's based on like 1 or 2 samples. And so honestly, like, I'm sending my technician to Africa in January, and she's going to stay till April to answer that question specifically. And she's also going to do some feeding assays to see if the insects are preferentially attracted to carcass-grown plants and not. And so I think there is, but we just don't have enough data yet to say. And also because identifying African grasshoppers, we've discovered we have to do it genetically. Like, there's just no morphological text out there to help us do it. So there's also a whole sequencing component that has to go into this. Evan.
I've been like struggling how to phrase this question. I think about it throughout the talk. But there seems like there are probably potentially some dynamics where the movement of the animals, or their consumption, or where they like spend time as fish, or potentially the elephants, where they're living and eating, consuming, and like building that nutrient base throughout their lifetime and then dying, might be, there are biases in, or differences in the locations of those two. You also then have a translocation of nutrients from maybe a bigger area to one specific area. Like, do we know about, like, do elephants go somewhere specific to die and eat other places? Like,
They do. So I'll say, so the question is, do elephants tend to go somewhere specific to die, thinking that those specific places might then be the hot spots where all these nutrients are being translocated and dumped to in a concentrated manner, right? So the answer is, the idea of an elephant graveyard that might have popped up here and there is like mythical. There's not one single spot where elephants will repeatedly congregate to die, unless we do it, and then we dump all their bodies in a single spot. I will say the big pain in the ass of this project is so the main cause of elephant mortality in the park is old age, because elephants don't replace their teeth, and so by the time they're 60 or 70 years old, they've ground their molars down and they don't have any way to eat, and they literally starve to death. So if you're an old starving elephant, where's the easiest direction to walk? Downhill. And where do you want to die? In a stream. And so a lot of the carcasses we actually can't work up because they wind up being in pans, in streams, and in other water bodies where the elephant has just like moseyed downhill to die. And so there's a huge bias there, and it's one we're not capturing because we literally cannot, and for hippo reasons, will not do this work in a stream.
Well, go ahead, Lauren.
Yeah, this is sort of I feel like I'm on shaky ground asking a question like this, but is there any rationale for considering the grasshopper gut symbionts as part of the microbial community, and that if you include that, then you actually have sort of more stable microbial communities?
So the question is, if we talk about grasshopper gut microbiota, is that the reason we're sort of seeing more stable communities? And the answer is that is literally one of the things Jane and I think is happening. And one of the grad students I was in my lab briefly was actually doing that and extracting the guts for us to sequence so we could answer that question. We just haven't got there yet. But that's the original motivation for this whole study was grasshopper pooping not just the nutrients, but the actual microbiota into the soil.
Although oftentimes gut microbiomes have a really hard time living outside the stable life of a gut. So.
And especially grasshopper gut microbiomes
Yeah.
like tend to be pretty distinct and identifiable. Like you more or less know what families and genera are going to be in there.
Yeah, but alrighty. Well, if we don't have any more questions in the house, we have the opportunity for people on campus to come to lunch and chat more with Nate. I know there's more meetings throughout the day, and we also have, of course, happy hour at 4 p.m., so please join us. And to everyone online, thank you so much for attending, and thank you so much. One last round of applause for Nate.
Thank you.